Greetings everyone and welcome to Mr. Cheese channel. Today we are performing a surgical deep dive into the Northwest Provincial Grade 12 Life Sciences paper from March 2026. We are focusing specifically on the core mechanics of genetics and human reproduction found in Section B.
I am so ready for this, Alex. I have been looking at the diagrams for question 2, and they look a bit intense. Can we start right at the beginning with 2.1.1? What are we looking at in those meiosis diagrams?
Of course, question 2.1.1 asks us to identify three structures, X, M, and Z. Looking at the marking guidelines, X is the cell membrane, which is that outer boundary holding everything together. M represents a chronotid, which is one of the two identical strands of a replicated chromosome. Finally, Z identifies the spindle fibers, those protein threads that pull the chromosomes apart.
That makes sense. It's like the machinery of the cell. Now, 2.1.2 asks us to identify the phase in diagram 1. They're all lined up in the middle, but they're in pairs. Is that important?
It is crucial. Because they are lined up in homologous pairs at the equator, this is metaphase 1. If they were in a single line, it would be mitosis, or metaphase 2. This brings us to 2.1.3, which asks why pair Y is considered a homologous pair. The markers are looking for three specific observable reasons. They have the same shape, the same size, and the same centromere position.
I see. So they're like matching socks. In 2.1.4, there's a process happening at Y where they're touching. What is that called?
That is crossing over, and it is a massive part of why we are all genetically unique. To get full marks here, you have to name it and then describe it. The homologous pair of chromosomes lie next to each other. The chromatids overlap at points called chiasmata, and they exchange genetic material. In the marking guidelines, naming crossing over is actually a compulsory mark.
Wow, so you can't miss that term. What happens after that? 2.1.5 asks to describe the phase that follows diagram 1.
Since diagram 1 was metaphase 1, the next phase is anaphase 1. In this stage, the homologous chromosomes are separated because the spindle fibers shorten. As a result, the whole chromosomes are pulled toward opposite poles. This specific separation process is officially called disjunction.
Disjunction. I will remember that. Now, 2.1.6 asks for a table comparing mitosis and meiosis. I know they produce different numbers of cells, but what else does the memo require?
You need at least two differences and a proper table structure. For mitosis, two identical cells are produced, no crossing over occurs, only two cells are formed in total, and the purpose is to form gametes or sex cells.
That clears up the cell division part perfectly. Let's move to 2.2, the female reproductive system. 2.2.1 is about the letters. Where do the sperm go and where does the baby grow?
According to the diagram and memo, sperm cells are introduced at D, which is the vagina. The embryo is implanted at C, which represents the endometrium or the lining of the uterus.
And 2.2.2 focuses on part A, which looks like the fallopian tube. How is that adapted for its job?
Part A is adapted in a few fascinating ways. It contains cilia, which are tiny hair-like structures that provide a one-directional beating to ensure the zygote moves toward the uterus. It also has peristalsis, which is involuntary muscle movement that helps push the zygote along. You only need to explain one of these for the 2 marks, but knowing both is better for your understanding.
Let's keep it moving. Looking at question 2.2.3, there's a diagram of the female reproductive system with cancer spots marked on the fallopian tubes. The question asks if it's still possible for this person to get pregnant. It looks pretty serious, so I would guess no.
It is a common trap, Sam. If you look closely at the diagram, the cancer spots are only indicated on one side. The marking guidelines for 2.2.4 clarify that because the other ovary is still intact, it can still produce an ovum. Ovulation will continue and fertilization can still take place in the remaining healthy fallopian tube.
Oh, Oh, that makes sense. So as long as one lane is open, the process can still happen. Following that, 2.2.5 asks to describe oogenesis. That's the actual production of the egg, right? How does that work, step by step?
Exactly. It starts with diploid cells in the ovary undergoing mitosis to form numerous follicles. Once a female hits puberty under the influence of FSH, or follicle-stimulating hormone, one cell inside a follicle enlarges and undergoes meiosis. Out of the 4 cells produced during this meiosis, only one survives to become a mature haploid ovum. This happens like a clockwork in a monthly cycle.
Only one? That's a high-stakes competition. Moving to 2.3.1, we have a bunch of animals labeled A through F. The question wants to link them to reproductive strategies. For example, which ones show external fertilization with precocial development?
For that one, you're looking at B and C, which are the frog and the fish. Precocial means the young are relatively mature and mobile from the moment of birth or hatching. Now, if we look at altricial development in 2.3.1b, where the young are born helpless, the answers are C, D, and F.
Wait, C is there for both?
Yes, some fish species have varying levels of independence. But notice for vivipary in 2.3.1d, which is giving birth to live young, the answer is F, the human embryo. Humans are the classic example of internal development where the mother provides all nutrients directly.
I see. Let's switch gears to the molecular level. Question 2.4 is all about protein synthesis. 2.4.1 asks us to identify structure E and molecule X.
Structure E is the nuclear membrane, which acts as the gatekeeper for the nucleus. Molecule X is the double-stranded DNA itself. The paper then asks us to describe translation, which is stage 2 happening outside the nucleus.
That's where the code actually becomes a protein, right?
Spot on. During translation, each tRNA molecule carries a specific amino acid. The tRNA has an anticodon that must match the codon on the mRNA strand. When they match, the tRNA drops off its amino acid at the ribosome. These amino acids are then joined by peptide bonds to form a long chain which eventually folds into a functional protein.
I see some base sequences in 2.4.3. If the DNA sequence is AAA, GTA, CTG, CGC, what would the mRNA, molecule Y, look like?
You just have to remember your pairing rules, but remember that mRNA uses uracil instead of thymine. So AAA becomes UUU, GTA becomes CAU, CTG becomes DAC, and CGC becomes GCG.
And for 2.4.3b, what would be the anticodon for that second triplet?
The second triplet in the DNA was GTA. The mRNA codon we just made for it was CAU. Therefore, the tRNA anticodon that matches that mRNA would be GUA. Notice it's almost identical to the original DNA, just with uracil.
That's a great shortcut. Finally, let's look at question 2.5. It's a forensic mystery about a deceased soldier. We have DNA profiles for the soldier and 4 sets of parents. 2.5.1 asks which ones are the actual parents.
To solve this, every single band in the soldier's DNA profile must match a band from either the mother or the father. If you track the lines across, you'll see that all the deceased soldier's DNA bands correspond perfectly with the bands from parents C and D.
It's like a biological barcode. 2.5.3 mentions other uses for this technology. I know about crime scenes, but what else?
The marking guidelines list a few vital ones: paternity testing, diagnosing hereditary diseases, and even establishing matching tissues for organ transplants to ensure the body doesn't reject the new organ.
This has been incredibly helpful for visualizing how these concepts actually show up on the exam.
If this helped you, share it with your followers, and also follow Mr. Chi so you never miss out on these deep dives. Comment below what you would like to see us break down next, whether it's more life sciences or another subject entirely.